I really do feel like this is an honor for me to talk to you about something that is really important to me, and the reason that I came to work for Lindamood-Bell at all, and that has to do with the strong and abiding interest that Nancy has in research, so much so that, unlike most companies, she has a department of research and development, and I'm very proud to be part of that department. So I'm going to do a very Lindamood-Bell thing and set the climate as we start. I want to talk to you about these two very important images, brain imaging studies that have come out that show the effects of our instruction on reading skills and the concomitant brain changes. Now, I think this is particularly important because these are scientific studies that back up what we do every day at Lindamood-Bell. This is how we present ourselves to our public, to the world. It's through our publishing. And the studies that I'm going to present to you are particularly important in that regard because they were independently designed and executed. That is to say, I was involved in these studies, but I wasn't an employee here, nor were any of the people who actually carried out the measurements and the design of this study involved with Lindamood-Bell. That's really important. That means that the results you can take to the bank. There's no conflict of interest involved. The other thing is that these studies have been vetted by peer review and published in journal articles that are highly respected ones. They were published in, respectively, Neuron and NeuroImage. The new one is coming out in NeuroImage. These are the journals that are among the top ones that scientists who study reading read and publish in. So this is not middle of the road in any sense. This is very high caliber science that we're talking about, and the results that we're talking about have been measured statistically. So we're not just saying we get good gains. We're not just saying that we're based in research. We're saying we're based in evidence, and I think that that's a theme that we've been hearing, and that you're going to see in the center's report that comes out. We are evidence-based in this company. So let's go ahead, and I have to say, Nancy, I am not nearly as good at these buttons and whistles as you are. You make it look really easy, so I'm not going to do a lot of switching back and forth. You're not going to see my face for a while, and you needn't, because we're going to have some slides, and although I know you're all very good at picturing things, I will help your picturing with some diagrams. So we're going to start with a short lesson in brain anatomy and some terminology. We always need to have our terms in place, and even though these may be words that you are familiar with, let's see if this works. Okay. They may be used slightly differently in this context. So the way the talk will be organized is with general and specific brain anatomy. So we're going to start with the whole brain and then work our way down to finer structure. Then we'll talk just a little bit about structure and function as we apply neuroimaging to get that information. And then finally, I will talk about those two studies that I've already referred to and tell you about the changes that we got in those studies, and impress upon you, I hope, how important they are. And I do hope that you will read these papers, because you're going to be armed with the information that you need, and you'll find them very interesting, I think. And they're not very long either, so it's pretty good reading. So we'll talk then about some of the terms that are used in brain anatomy. These are listed for you here, and you can read them. I won't read them to you. Some of them you may be familiar with and some not, but we will go through some pictures and get oriented to them. So in this first one we see a picture of an actual brain. This is what we might call a fresh brain because it hasn't been fixed in a solution for study yet. So it's still kind of pink. It is pink because it's highly vascularized. You can see see if my pointer actually does work up there. Whoops. Well, I thought I could use a mouse at least. Okay, there's the pointer. Can you see it?
Yeah.
Yeah, all right. We tried to make it red, but we couldn't manage to do that for some reason. So, dorsal is the top of the brain. Ventral is the bottom or underneath side of the brain. And, of course, anterior and posterior are obvious. So this brain is, this is the left side. It's pointing towards your left. That's the anterior part. Typically, then, the eyeballs would be right down here. This whole thing is the brain. This big part here that looks sort of like a boxing glove is actually called the cerebral cortex, and it's kind of like it has a thumb here and a fist up here. And in fact, just like with a glove, if you moved this thumb part away from the rest of the brain, you would be able to see that there are more of these convolutions and more of the vessels down inside this very deep crevice. This part of the brain back here is called the cerebellum, and it's involved in memory, in motor memory, acquisition of motor, sequential motor planning. So it's very important for walking and balance. And together the whole thing is the brain, and of course then it extends down to the spinal cord underneath. And that's been severed in this picture. Now we've turned the brain to the inside, and this is called the medial aspect or medial view of the brain. And you can see that there are yet more and more of these convolutions, hills and valleys, all around the inside, the medial view. And down here we can actually see the ventral side of the brain down at the bottom here. So there are lots of these, and it's highly convoluted. And we're looking just at one hemisphere. There are, of course, two, and if we sliced through the middle of them, we would see this structure here. This is the corpus callosum. It's made of fibers that run between the two hemispheres so that they can talk to each other. So if they weren't talking to each other, we would have a hard time coordinating our activities. This is how we are able to put together information and take that information and turn it into motor responses. I'm going to try to remember always in these to show you the anterior part so you know which way the brain is facing. If you're not used to looking at brains, sometimes that can get confusing. So back again, we're looking at the lateral aspect of the left hemisphere, and we see that we can divide the brain now into some other portions. These are called lobes, just meaning areas, and each one of them has a name. So the frontal lobe is in the front, conveniently, and the temporal lobe is this green part down here. The part that's been colored yellow here is called the parietal lobe, and this region in the back that's sort of pink is in this picture is called the occipital lobe. The odd thing about the brain, the way the brain is organized, is that even though the eyes are up here in the front, the information that comes directly from the eyes into the brain goes all the way to the back of the brain. Very strange sort of organization, isn't it? But that's the way it's set up. And so the first sensory information that we have that comes into the brain is at the occipital lobe. in the very posterior part. I'm losing my mouse here for some reason. I'm not sure.
Yeah.
Is that what I need, a pad? Because I keep losing my arrow. So, sorry about that. So, here it is again. I don't know what happens. But so here is, it's in this region, down in this crevice, where the information about what we hear comes in. So, in fact, the whole back of the brain is very sensory. Sensory information comes in from the world in the back of the brain, and in front of this deep crevice is the output part of the brain. So we have input in the back and we have output in the front. We can subdivide these lobes even further. And when you read these papers, or any paper about brain imaging, you will see these terms used. And as soon as you read this in a paper, you'll know exactly, you'll be able to picture exactly where they're talking about. And that becomes very helpful if you're going to understand how we know what we know about reading and any other activity that we care to study. So just to give you an example, these bumps and ridges are called gyri, or one is a gyrus, and each one of them has a name. Up here in the front we have a gyrus in the frontal lobe, and it's in the middle of the frontal lobe, so it's called the middle frontal gyrus. And similarly, in the superior temporal area, there's a superior temporal gyrus, and it's demarcated by this long crevice here, this long sulcus or valley, and the superior temporal sulcus. So, in addition to that, some of these areas have their own special names. Right back here is an area called the angular gyrus. That's important for reading this region back here. It's in the inferior parietal lobe. So we look at this cartoon of the medial view. We're not going to talk much about the structures on the inside. There's the corpus callosum again, the cerebellum again. But I do want you to make a picture now of the precuneus, this area that's circled in red. That is an infolding of the parietal lobe that goes into the medial side of the brain. And that's also going to be very important. So far you have something that you need to keep a picture of and remember, and that's the precuneus in the posterior part of the brain, in the sensory area, and it is part of the parietal lobe. Now we can actually talk about it in even more fine detail by looking at these areas that are named by Brodmann, who subdivided each of these gyri and sulci into even more finely grained areas. And this was done on the basis of microscopic structure. So he looked at the structure of these different, within these different areas, even within their like sub-regions. And he gave them numbers that are based on those microscopic features. So we get more and more specific. The arrow is pointing to, I don't know if you can tell, but it's the number 39, Brodmanns area. So we can look at that area, and we can...
It's on there.
Yeah, okay. We can look at this area, and we can give it a name. If I had a classroom here, I would ask them what they would call this area. But I won't embarrass anybody in this room. It's in the inferior parietal lobe, not the superior part, but the inferior part. It's also in the angular gyrus, and it's Brodmanns area 39. So this is a very specific address. See, us brain people, we love this stuff. And we've just been looking at the outside layer, the gray area on the outside that's made up of nerve cell bodies. But we can look at a cutaway here and see that the gray matter is just a ribbon around the outside. It's like a wrinkled gray sheet or covering on the outside. It's actually very thin. In some places it's less than 1/10 of an inch thick, and in others maybe as much as 1/4 of an inch. So it's not very much, and yet it covers a great deal of territory because of all of the enfolding. We call it gray matter because it does look gray when it's been fixed in a solution, and we call the inside of it the white matter because it looks white, and that is because of the connections between these gray cells being covered with a fatty material, and I'll show you that in just a moment. So here's the second thing I'd like for you to picture. And remember, and that's the location of the hippocampus. The hippocampus is not visible from either the lateral, ventral, or medial aspects because it's buried down deep in the temporal lobe. And so this cutaway, though, does show where it is. There's a hippocampus in each hemisphere, so you have a left and a right. So take a picture of that and recall it later. This is what a typical neuron in the brain looks like. The important parts of it are the cell body. These feelers out from the cell body, called dendrites, they receive information from the environment in chemical form. So chemicals, or neurotransmitters they're called, are picked up in all of these areas, and there are many. Often there are just thousands and thousands of these, and we think that when nerve cells are involved with each other, that these trees, these dendritic trees, grow and expand. They take up more space, which is probably why we would expect that with training and learning and during development, we would see changes in gray matter volume. So those are two important parts of the nerve cell. Another is this extension that goes out, and it may go for a very long distance. It's called the axon, and it is the way the nerve cell then reaches out to the next nerve cell in the line and gives information to that nerve cell. So the chemistry comes in here. It causes an electrical impulse that discharges down the length of the axon, down to the axon terminal, and chemicals are then released there, and that is the message that is sent to the next neuron. So I'm going to just show you a little clip here that shows how that works. Chemicals at the front end, the nerve impulse going down the axon, and then a release at the other end. These are the ways that our brains work. So your brain is working on chemistry and electricity at all times. This picture from the NIH shows how two actual nerve cells would be communicating with each other. And you can see that there are lots and lots of dendrites on these cell bodies. So here are the bodies. These are all dendrites, and they branch, and they have finer and finer branches. So they can communicate with literally hundreds of other nerve cells at one time. They send their message down the axon to the next cell, and so on. So these are very complicated, very involved neural networks. Well, we've talked now about gross anatomy and finer tuned anatomy and our nomenclature. I'd like to talk to you a little bit about how we know what the brain does. And it's only been fairly recently that scientists have known what the brain looks like when it's behaving, what a normal brain can look like. because we have only recently, in the last few decades, had machines like this MRI magnet that you see that could tell us that kind of information. It gives us these wonderful pictures that are very crisp and clear. They look so much like the actual brain, and through the miracle of computer reconstruction, we can actually look at sequencing slices taken through the brain at any angle. We can reconstruct whole structures. We can take measurements of structures. We can take measurements of the volume of the different tissue types, the white matter, the gray matter, and the cerebral spinal fluid that is in the spaces that are in the middle and around the outside of the brain. And I meant to mention, because I think this is really interesting, I forgot to say it before, that the brain is literally floating in this fluid. It's suspended in it, so that the brain is never touching the bony structure that's around it. The skull is there to protect the brain, but then you have to have something to protect the brain from the hard structure, and that's the function of that fluid. It also cushions the brain if it's jarred. So until we had an MRI, we had to just guess at what the brain was doing based on loss of function. So 100 or 200 years ago, if somebody wanted to understand more about how the brain works, they would observe what kind of function was lost when a person had some kind of injury to their brain, like an actual injury or a stroke. And if they lost some function, then they would look to see where that stroke was and say, well, that area must be responsible for that function. So a scientist named Deletion in the middle 1800s noticed that people who had a particular kind of insult to their brain lost the ability to read. He called that alexia. People with alexia are able to see and hear and talk and write, but they cannot read. They lose that particular function. And they can't even read the words that they've just written themselves. So it was quite a curiosity at the time that it was first observed. Deletion and others have subsequently looked at a number of stroke and injury patients to try to figure out where reading might be in the brain. And they came to some conclusions, not always in agreement with each other, about where that might be. But now we don't need to guess. We have the MRI. We can use it to make these wonderful images, and we can do this without radioactive substances and without injuring the brain or invading it at all. There have been literally hundreds of studies of brain activity, all kinds now of activities, and cognitive in particular, but also motor activities, various types of cognitive functions in all ages, because we can scan children who, as long as they can stay still in a scanner, we can do imaging, and we can look at the function of the brain, and we can also look at the structure of the brain using the MRI magnet. A number of these studies have involved reading or reading processes. Most of what we know now has been based on phonological types of tasks, but increasingly we and others are interested in higher level function, and we will shortly be beginning a study that looks at comprehension in autistic brains. For the moment, I'd just like to show you three areas that are typically found to be active during reading in the typical brain. And usually are unactivated or at a lower level of activation in the brains of people who don't read well. Now, the left posterior inferior temporal cortex, where would that be, do you think?
On the left side?
On the left side. Good answer. Right back here in the back, just as it was described. This is often called the visual word form area because this is where whole words seem to be processed. And then there is the temporal parietal area. Notice that we've joined together two terms, temporal and parietal, because this section is quite large and it takes in both temporal cortex on the temporal lobe and parietal cortex in the parietal lobe. Another area that we'll focus on is the inferior frontal gyrus, and that's, of course, in the inferior frontal gyrus, exactly where it says it is. So this visual word form area is responsible for orthographic processing. This is the place that becomes more active increasingly as people become more automatic in their recognition of words. In the temporal parietal cortex, information from auditory and visual sensory systems is integrated, and this is where decoding takes place. So this is phonological assembly area. There's some memory involved in activation in that area, and Marcel Just and Nancy Minshew and the folks up at Pittsburgh have identified an area right in the very top part of that, right up in here, that is activated when people are imaging sentences. So that's very interesting to us too. This frontal area, remember I said, was output, and this is the region that is particularly devoted to verbal production. It is the area where putting together the sounds that are the motor assembly in your body is going to need to produce a word, so it's important to us for reading. Okay, so here is the sum total of your physics lesson for today. This is how it works. This is how we get the information from the brain out to these wonderful pictures using a functional MRI. When nerve cells are active, just like muscle, when muscle is active, they demand more oxygen. So the blood supply with oxygenated blood goes to those active areas. Functional MRI is sensitive to differences between blood that has oxygen and blood that has released its oxygen. And it is through that sensitivity that functional MRI measures activity. So it's really indirect. But functional MRI is really measuring blood flow that changes in blood flow that are responding to increased demand as neuronal activity is increased. And the equipment these days is so very good and so very sensitive that we can measure just millimeters of area where activation is taking place. It's really pretty remarkable. This study from Guinevere Eden's lab shows how children who are counting tall letters in words activate their brains. So children in the 6 to 9 year bracket use some of this posterior temporal area. As they become better readers and reading becomes more automatic, words become more automatic. Even though they're not actually asked to read the words, but merely to look at them, they can't suppress actually reading the word. And so additional areas come into play. So in addition to that area, also a parietal region and a frontal region have come in. And by the time you get to be a young adult, you can see that activation is more widespread as reading has gotten to be more efficient. That study also showed us that over time, as reading skill improves, areas in the right hemisphere, these golden areas, increase in their activation at the same time that areas in the right hemisphere are decreasing their activation. So the brain is really becoming much more specialized. This tells us that reading is one of those complicated functions that takes a long time to develop and that actually differentiates the hemispheres. Now I'm going to talk about the first study that was published in Neuron in 2004. And this was a collaboration between Wake Forest and Georgetown. We were looking at adult dyslexics, and these were dyslexics who had been identified in childhood, and they were quite severe. They still had problems as adults. And we were asking two questions. We wanted to know, what does the brain look like in a dyslexic person? How does it differ from a typical reader when they're doing a phonological task? And then beyond that, and even more important to us, was what changes, if we can change their reading skill and make them better readers, what changes in those brains? The overall design was this. So we had an assessment. This was a regular battery, the kind of thing that you do every day in the centers. And we did functional MR. And then 8 weeks of Lindamood-Bell instruction took place. Following that, there was a second assessment and a second MRI. We didn't do the instruction. Lindamood-Bell did that instruction, and D.C. DeRoe was involved in that. She was there for the whole 8 weeks and taught these adults in 3-hour sessions, 5 days a week. If you can imagine being an adult and doing this. These people had jobs, and they worked around their jobs so that they could come to their lessons, and they were wonderful students. They were highly motivated, as you can imagine. The folks who got this were just half of the dyslexic subjects that we had. So we divided the group in half, and half of them got the instruction, and half of them did not. During that period, then, we were looking for changes in dyslexic brains that would take place as a function of the stimulation they were getting when they did LIBS, VV, and Seeing Stars. It was whatever they needed. It was a needs-based instruction model. And after they finished, we wanted to be able to tell the difference between what changes in a dyslexic brain. Some previous studies had looked at dyslexics compared to typical readers, and that's of some interest, of course, but we took a slightly different view and decided that we wanted to know what the dyslexic brain is doing. So during the task, or during the functional MR, we had to give the brain something to do so that those cells would be demanding oxygen. So we did a task that was a kind of typical phoneme deletion task. We first of all had them focus on a square, a computer screen that had a plus sign in the middle like this. So they were just looking during that fixation time. Then we gave them one of two tasks. Either they listened to a word, they didn't see the word, they listened to the word. They spoke the word that they heard. So in this condition, they were looking at the plus sign and speaking. Then we gave them another task, and in this task, we asked them to listen to the word, take away the first sound, and tell me what's left. So again, they were looking at the plus sign, they did a phonological manipulation. They took away the first sound and they spoke. So I think you can see here that the difference between the first task and the second task has to do with what they were doing to manipulate that phoneme. So we expected the brain to be showing us just that because we were subtracting out everything else. And what we saw was that these dyslexics increased activity in their parietal lobe. In fact, this is in Brodmanns Area 39 in the angular gyrus in the inferior parietal lobe, and that should be very familiar to you by now. Also, they increased an area in the ventral part of the posterior temporal lobe. And that was of some interest to us. But here's what we were really, really interested in. And that was, what did these dyslexic brains look like? When we compared what the profile looked like before the intervention and then after 8 weeks of Lindamood-Bell intervention. If we subtract again the two profiles, then what we have left should be what is the result of the instruction and what effect the instruction then had on the brain. And we found some very interesting things. We found that in the left hemisphere there were some areas that were increased as a function of the instruction in some of the areas that we exactly have been talking about. And in addition to a region down here that you can't see, which is why the dotted circle, it's down underneath, it's in the ventral part of the temporal lobe, and that's the fusiform gyrus down there. So we found increases in that region as a function of instruction. So in the right hemisphere, there were also a number of areas that increased in activation. Along with that, there were changes in their behavior. So we found differences as a function of instruction on a phonological awareness task, on the SI task. And in this case, we're noticing that the intervention group is the dark blue, and this is a statistical measure, a statistical outcome. So they are significant at the statistical level in their improvement over comparison to the non-intervention group. The same thing for decoding. We had a couple of different measures of decoding. And we also had differences in reading accuracy on the Gruy oral test. So we had, along with those changes in brain activity, we had changes in reading skill, which of course doesn't surprise any of us here. But this was something that was kind of new at the time because these were, first of all, adults. They were severely dyslexic, and we had identified them as children. And so we were absolutely certain that their problems were not acquired problems, but were developmental. They'd always been there. These skills also were not just in their ability to do phonological awareness tasks or word decoding tasks. They were differences also in reading in context. So we interpreted these changes in the left and right hemisphere as changes that were normalizing in the left hemisphere. So those were the product of remediation of their problem, and also areas of compensation in the right hemisphere. Now we come to the most recent and in some ways the most astounding study, I think, and that is this new study published in NeuroImage that shows gray matter volume changes as a function of reading intervention in a small group of dyslexic children. And here I want to also acknowledge a couple of people who were involved in the remediation part of this study. Lillian and Angelica were both very much involved in carrying out this study. We would not have been able to do it without their cooperation. But again, this was done independently of Lindamood-Bell, and the researchers involved had no connection with the company at all. So I press that because many of the studies that we've seen looking at intervention programs have not been independently done. So that's really important. I'm going to show you this cutaway again, showing the gray and white matter, just to remind you that what we're going to be looking at here is the volume in this gray strip around the outside. We want to see, and we predicted that we would see, some increases in some areas of gray matter that would be exercised during the instruction period. So we followed 11 children. They had only Seeing Stars instruction. We wanted to see just exactly what Seeing Stars instruction was doing to the brains. We knew we could improve skills using Seeing Stars, but we hadn't been able yet to show what that effect would be on the brain when it was used in isolation. So we took 3 measurement points in this case: before instruction, after instruction, and during a null period that was equal to the instruction time. That null period just means that there was no instruction during that period. And what we predicted was that there would be some gray matter increases that would go along with gains in reading performance. This is a figure taken from the paper, and this one is of reading skill changes. So time 1 to time 2 during the intervention period, all of the reading skills that we measured increased in had increases in score, and these were significant changes. During the null period, none of the changes were significant. So I hope you understand the importance of that. Between time 1 and time 2, there was an increase in skill. During the time between time 2 and time 3, those changes, those increases in skill level were maintained. This is very, very important. You've heard people say that anything that you do makes a change, especially makes a change in the brain, but will that change be maintained? So particularly we saw a lot of change in all of these different areas that we measured. I've already mentioned that. But in addition to that, there were 4 brain structures that showed significant increases in gray matter volume at the end of the intervention period, or the instruction period, as compared to the beginning. So time 1 to time 2 increases. And these are areas that are involved, importantly for us, in memory and particularly important in mental imagery. We're delighted with this outcome, of course. The increases also were maintained over time. So here is a figure from that paper. From time 1 to time 2, there were increases in all these areas in gray matter volume. So we're talking about structural changes now. Between time 2 and time 3, during the null period, there was no significant loss in those gains. It looks like there almost might be the beginning of some trends to continue to increase, and that's been shown in some other studies where increases in gray matter volume have been associated with some kind of learning or practice, but not with reading. So this is brand new. This is another figure from the paper, just to show you that in fact these were we can locate these places in the brain. This is a slightly different way of looking at the brain, so we're looking down on the top of it, and in fact in this one you can actually see the eyes. But it's easier if we look at these diagrams to point out where these places were. So I hope your visual imagery is helping you out here to show you, so you remember where the precuneus is. And this brain happens to be turned the other way, so this is the front. This is the anterior part. As I said before, it's part of the superior parietal lobule that's hidden down between the two hemispheres in this fissure that goes between them. You recognize this because you see the corpus callosum on the medial aspect of this brain. This area has several functions, the precuneus, but it is the things that are most important to us are the implications for visual imagery of letters, and it's also been reported in another study to be underactivated in dyslexic adults. So this is a really important finding. And then down here in the ventral aspect, and you'll remember this, I hope, from your mental picture, your imagery, that the fusiform gyrus had some, that we found some increased activity in the previous study after instruction in those dyslexic adults. In this study, we found increases in the gray matter volume in that fusiform gyrus on the left side. So that's really very interesting. And then the last place that we're having a particular interest in this study is the hippocampus. And we found active gray matter in volume increases in both hemispheres of the hippocampus. Because other people have found increases in gray matter volume when they have asked individuals to do some kind of practice, either motor practice or study of some kind, we expected to see some increases here and predicted that we would. And we think that this is because of seeing stars instruction actually induces generalized learning. So these are brand new. This is brand new, the first time that this has been found. Training-induced changes in gray matter were found in this group of children. There were reading improvements also induced by the intervention that were accompanied those changes. And particularly important, both reading improvement and gray matter volume changes remained over that period after the training ended. So these are the major findings from that study. So we can now come to some general conclusions based on what we've been talking about today. What we do changes behavior. What you do at Lindamood-Bell changes behavior. We've known that. That was not a surprise. What we do changes brain function and also changes brain structure. This is corroboration. This is big. This is really big corroborating evidence for what we do. The most important thing, perhaps, is that these changes make a difference that lasts. I mentioned before that sometimes people say that everything you do changes your brain, and certainly it does, but those changes don't all last. When you have learned a skill and you can hold on to that skill, that's what's important, and that's what we do at Lindamood-Bell. We change brains and we change lives. So I know you've heard it before. I will quote Nancy Bell and say, lucky us.